In today's discussion, I will basically discuss about two things. One is about the altered secretions of various types of pancreatic hormone, and another one is the synthesis, release, secretions, and mode of actions of glucagon hormone and somatostatin hormones. So in my previous lectures, we have covered the first part, that is about the insulin that we have discussed in the first lectures. So let me start with the rest of the part, that is remaining from insulin sections, or that is remaining from the beta cell part. So as it is mentioned here, the altered insulin secretions. We have discussed about the importance of sufficient amount of insulin secretions and how these hormones are secreted, how they act into the target cell. Now at the same time, we have to discuss about the impact of the altered hormone secretions or altered hormone release. Here's the topic of discussions is altered insulin hormone secretion. So regarding this, the insulin hormones, as we know, this is very, very important because it involves the maintaining of glucose balance, and at the same time, under certain conditions, what we have seen that there may be hypersecretions of these particular hormones or hyposecretions of these hormones. So both the conditions is not good for physiological point of view. So now let us start with what are the various impacts of altered secretions of insulin hormones. Here's altered insulin secretions. It refers to deviations from normal insulin productions. Generally, we know the most important type of cells that involves the secretions of these optimum insulins is the beta cells, and sometimes there is alterations of these optimum secretions. And here's due to these altered secretions, that may create two physiological conditions. The conditions are hypersecretions or hyposecretions. Both the conditions are very, very important in physiological aspect. Now see what are the most important consequences that may develop in these particular conditions. The most important ones is the development of a special conditions called the diabetes, right? And here's both the hyper and hypersecretions is related with the developments of the diabetes. And here as you can see, both the type 1 diabetes and type 2 diabetes may develop in altered insulin secretions. So what is the physiological basis of type 1 diabetes and how these insulin secretions promotes the developments of type 1 diabetes. See here, hyposecretions of insulin occurs in type 1 diabetes mellitus, where autoimmunodestructions of the beta cells leads to absolute insulin deficiency. So this is the most important characteristics, that in this particular case there is a destruction of the beta cells, and it is the autoimmunodestructions, and due to these autoimmunodestructions there is a insufficient insulin secretions or deficiency of insulin. And here's this insulin deficiency may lead to developments of some physiological conditions. So what are the physiological conditions that may develop in this insulin deficiency? You can see there is a developments of hyperglycemia and the productions of ketone bodies, as well as there is a high chances of the development of potential diabetic ketoacidosis, right? So what is the biological basis of this hyperglycemia? Obviously, if there is a deficiency of insulin, as we know, the insulin hormones is very, very important for the absorption of the blood glucose. So here, due to the deficiency of insulin, there is a less chances of glucose absorption from circulatory blood. It means there will be accumulations of the glucose in circulating blood. It leads to the developments of these conditions called hyperglycemia, right? At the same time, if you see the role of insulin deficiency in ketone body formations, yes, there is a rapid breakdowns of fats, right? And these fat breakdowns ultimately produce a higher amount of ketones. And in this particular case, what we see, there is a high chances of the depositions of the ketone bodies in circulating blood that ultimately builds up a acidic conditions of the blood, right? This is called diabetic ketoacidosis. So here's insulin deficiency may lead to the development of two special conditions, that is hyperglycemia and diabetic ketoacidosis. At the same time, if you see another important type of mellitus that is also related with the insufficient insulin secretions, that is the type 2. So what is the physiological basis of this? In this case, you see type 2 diabetes mellitus is a chronic metabolic disorders, and it is characterized primarily by insulin resistance. Yes, the most important things. So here's there is a sufficient amount of insulin is present in the circulating blood. Means the beta cells are producing sufficient insulin, but the cells, the body tissues are unable to absorb. Absorbed properly. On the other hand, we could say, though there is a sufficient insulin present in the circulating blood, it does not facilitate the absorption of the blood glucose, right? This leads to the development of these conditions called the resistance conditions. Here, it is characterized primarily by insulin resistance in peripheral tissue, mainly the muscle cells, adipose tissue and livers, and a relative insulin deficiency due to the progressive beta cell dysfunctions. It means here also you can see the dysfunctions of the beta cells leads to the development of conditions called insulin resistance. You can see here the two conditions. In the first case, both the case, you can see the beta cells are the most important responsible for the synthesis of insulin hormones. But in case of the type 1 diabetes, there is an immunoattack by the T cells, cytokines as well as the macrophages. So due to that, the beta cells loses its capacity to release insulin hormones, right? Here's there is a condition that develops called absolute insulin deficiency, and in this particular conditions there is a high chances of the development of pathological states called hyperglycemia. Now at the same time, in case of type 2C, the beta cells are producing insulins, but the most important features of this is there is a development of insulin resistance. Means cells are unable to produce absorb the glucose from circulating bloods, right? In both the case, the conditions develop. This is a hyperglycemic conditions. On the other hand, you can see sometimes what we see, there is a hypersecretions of these hormones, right? So these hypersecretions also not helpful for physiological systems. So how these hypersecretions of insulin affects? See, these hypersecretions of insulins, which is also known as hyperinsulinemia, it can result from insulinoma. So it is a special type of conditions. In this particular conditions, what we see, there is the development of cell mass, right? Or the development of tumors in specific cells, mainly the beta cells from our insulin are synthesized. And other than this, these conditions may develop due to exogenous insulin overdose as well as congenital hyperinsulinism in neonates. So here, in case of neonates, we can see another conditions may develop. So in both the conditions, what we see, there is a hypersecretion of insulins. Now, this leads to the recurrent hypoglycemia. In previous case, what we have seen, so hyposecretions of insulin promotes the development of hyperglycemia. But here, hypersecretion of insulin promotes the hypoglycemia, right? And other than this, you can see these hypersecretions also affects our various types of metabolic process, right? So what are the other things that may be affected by the hypersecretions of insulin? See, chronic hyperinsulinemia in the setting of insulin resistance may contribute to the development of some other conditions like the atherosclerosis formations and hypertension and in dyslipidemia. So these are some metabolic states. These metabolic states are influenced by the altered secretions of insulin. Mainly here's the hypersecretion of insulins, or we could say chronic hypersecretion of insulin promotes the development of this metabolic state. Yes, other than this, certain medications and functional certain medications also affects the proper insulin secretions, and these medications also promotes the development of certain types of disease that is related to the metabolic state of the body, right? Thus, we could say the altered insulin secretions plays a central role in metabolic disorders, especially those related to glucose homeostasis and energy regulations. So this is what is the importance of having the optimum amount of insulins. That is the importance of the beta cells, proper functions of the beta cells. Now, like this, the altered secretions of insulins, we will discuss about the altered secretions of glucagons and somatostatins too. But before that, we have to discuss about the synthesis, secretions, mode of actions of both the glucagon hormones as well as the somatostatin first. Then we will discuss the altered characteristics of both the glucagon and somatostatin. See here, the next part is about the synthesis of glucagon. As we have already mentioned that there is certain types of cells that is basically involved with the secretions of this type of hormones. Here, in case of glucagons, the most important type of cell is the alpha cells, right? So here, and obviously it is present in the islets of Langerhans, and during the synthesis of these glucagon hormones, what we see at the initial point of time, it was found as a pre-pro-glucagons, which is the inactive form of glucagons, right? So to become an active hormone, it must undergo through a specific cleavage steps to become an active form of glucagons. So here, if you see the characteristics of the pre-pro-glucagons and the active form of glucagons, you'll find some differences. In the alpha cells, pre-pro-glucagons is flipped to pro-glucagons. Yes, like previous ones, there is a cleavage takes place, right? So it means that shortens the chain length, polypeptide chain length. And ultimately, the pro-glucagons undergoes further enzymatic cleavage to form the active form of glucagons. So, in most of the cases, if you see the number of amino acid sequences that is present in the glucagon structures, that is maximum of 24 to 29 amino acids. In most of the cases, 29 amino acids are present in the active form of glucagons. Here's other pro-glucagon derived peptides are produced in the intestines. Means the alpha cells, these alpha cells are not only the sources of the active form of glucagons. If you see there are some molecules, there are some hormones which looks like a glucagons that are produced from other parts of the bodies, like intestines produce some pro-glucagons, that is basically the peptide. Now, what is the exact mechanism of this synthesis of glucagon hormone? Obviously, for the synthesis of this glucagon hormone, there must be a specific gene. That is, here's the GCG gene that is specific for the glucagon hormone synthesis. And the transcriptions of this glucagon specific genes produce the pre-mRNA, and the processing of this pre-mRNA produce the mature messenger RNA, which will undergo with the translation process, right? So these translations actually takes place in the rough endoplasmic reticulums, and it produces the pre-pro-glucagon first, right? So this is what is mentioned here, pre-pro-glucagon, right? From where it is synthesized after the translations of this gene, right? And after these formations of pre-pro-glucagons, it passed through some cleavage events, right? So here there is a cleavage of signal peptides that takes place in the endoplasmic reticulums, and it forms the pro-glucagon first, right? And these pro-glucagons again transport to the Golgi apparatus where packaging takes place, right? And this packaging is basically carried out by certain types of enzymes, and finally it forms a mature active form of glucagons having 29 amino acid sequence. Now once the mature form of glucagons are formed, the next thing is it must it must reach to the circulatory blood first, then the target site. So here you can see first is stored in the granules, and based on certain stimuli, the glucagon hormone are released, and ultimately it is exocytosed into the bloodstream. Now this is the basic the flow of the glucagon hormone synthesis. So obviously each and every different steps are important for the synthesis of the active form of glucagon hormones, which is having a certain number of amino acid sequence. You can see the chain-like structures of the glucagon hormone. Here, so one is N-terminal ends and one is C-terminal ends. Now, the synthesis already completed, and it is already now the hormones are in the bloodstream, right? Now the hormones are in the bloodstream. So, what next? Next is about the most important things. What are the most important factors that regulates the glucagon hormone secretions, or what are the stimuli that regulates the hormone secretions? There are certain physiological conditions that either promotes the secretions or inhibit the secretions of the glucagon hormones. You can see what are the most important influencing factors, like hypoglycemia. As I've already mentioned the terms, the hypoglycemia is very much related with the hypersecretions of insulin hormone, right? So it means when there is a hypersecretion of insulin hormones, when there is a hypersecretion of insulin hormones, it's somehow linked with the secretions of glucagon hormones. How? See, when there is a low level of blood glucose, these conditions triggers the glucagon release, the first conditions. Now, in another way, we could say when there is a higher secretions of insulin, it will promotes the, it will triggers the, or it will somehow lead to the glucagon release. And at the same time, if you see the plasma amino acids, so elevations of the plasma amino acids also influence the glucagon hormone secretions, especially arginine and alanine amino acids. That's the most importance for the glucagon hormone secretions or glucagon hormone release. Some sympathetic stimuli also promotes the glucagon secretions, mainly the activations of beta adrenergic receptors by some catecholamines like epinephrine and norepinephrines under certain conditions promotes the secretions of glucagons. So what is the conditions? The conditions is the stress conditions, exercise states, and hypoglycemic states. All these states promotes the secretions of glucagon by activating the sympathetic activity of our systems. Other than this, cholecystokinin also stimulates the glucagon release. So here it means there are some factors that promotes the glucagon release. Now, how these glucagon hormones are acts to the target cells, or what are the actions that is carried out by this glucagon hormone? Here you can see, as I mentioned, these are the factors, or these are the stimuli that facilitates the hormone release. Now, so once these factors are available that facilitates the release, that not only facilitates, that directly not facilitates the glucagon hormone release. Instead, we could say they alter some mechanistic process of the cells. What are the changes that takes place during the glucagon hormone release? You can see the first steps. The conditions was the low blood glucose. The conditions was certain type of amino acids promotes the glucagon secretions. The conditions was sympathetic activations or stimulations. So that particular conditions activates the alpha cells of islets of Langerhans, right? So activations of the alpha cells alters some ionic balance of the alpha cells. How? Once alpha cells gets activated, it affects the potassium channels that is present in the alpha cell membranes. Generally, though there is an opening of the potassium channel, but what we see is in this particular case, the efflux of potassium is little less in comparison to the case that was found in case of insulin actions, right? So there is an efflux of potassium ion, but the amount of efflux of potassium is less. So this efflux of potassium also allows this activation of the alpha cells also allows the influx of some other ions, mainly the calcium and sodium ions, right, with the influx of potassiums. So here in this particular stage leads to the changes in the membrane potential or alpha cell membrane potentials. So this membrane potential change are nothing. This is the polarization change called depolarizations. So here's the depolarizations of the alpha cell membranes as well as the excessive amounts of calcium ions. Both the things promotes the vesicular movements towards the alpha cell membranes, right? In this vesicles actually nothing. Vesicle contains some granules, and these granules are actually the glucagon hormone. You can see depolarizations of alpha cell membranes leads to opening of voltage gated calcium channels that causes the influx of calcium ion, and this calcium ion triggers the exocytosis of the granules, and ultimately there is a release of glucagon hormones in the extracellular space. And once these hormones are released to the extracellular space, it is diffuses to the blood capillary, and ultimately it enters to the bloodstream. As I mentioned, once the hormones reach to the bloodstreams, the next thing is it must reach to the target site for its actions. So here it is transported to the target site. The most important target site for the glucagon hormones is the livers. So now we'll discuss about how this glucagon hormones affects the liver or hepatic tissue. So this is what is the exact flow of how these hormones are synthesized and released. Now there are some molecules which inhibits the secretions of this particular glucagon hormones. So what are that conditions? Like hyperglycemia. And what I have mentioned in previous slides, I have mentioned in hypoglycemic state is the state which promotes the glucagon secretions, but these conditions inhibit the secretions or suppress the secretions. Along with this, you can see other hormones also equally important to maintain the balance of glucagon. Here mainly both the insulin and somatostatins inhibits the secretions of glucagons along with the hyperglycemic conditions. So these are some factors or stimuli, how this stimuli affects the secretions of glucagon. Now, what is the exact mechanism of this hormone action? As you already know, this is a type of peptide hormone, right? So obviously it will act to the second messenger systems, right? Because both the glucagon hormones, insulin, they all are peptide hormones. You can see what is the exact natures of glucagon hormone actions, or what is the mechanism of actions. Obviously there will be a specific type of receptors called glucagon receptors, and these glucagon receptors is also known as the G protein coupled receptors or GPCR, and it is predominant in the liver cells or hepatocytes. As I have mentioned in previous slides, the target site is the liver. So here's the activations of receptors stimulates the adrenal cycles via the G stimulatory proteins. So this is also discussed in numbers of sessions, and that stimulates the adrenal cycles, and adrenal cycles converts the ATP to cyclic AMP, and ultimately this cyclic AMP activates the inactive form of protein kinase. So up to this we already discussed in previous many lectures. Now see, as I mentioned, it will act in target sites. So what is the exact functions of these glucagon hormones in the target site? See, once the protein kinase gets activated, it shows several impacts. So what are the most important features of this activated form of protein kinase A or phosphorylated form of protein kinase A? It activates the glycogen phosphorylase, the most important enzyme, right? Most important enzymes. For what? For the breakdown of stored glycogen. It means we could say the glucagon is the most important one, which stimulates the glycogen breakdown, right? So how? By producing certain types of enzymes. So what is the enzymes here? This is the glycogen phosphorylase enzymes. And how these glycogen phosphorylase enzymes are produced? It is produced under the influence of activated form of or phosphorylated form of protein kinase A. At the same time, if you see the role of this phosphorylated form of protein kinase A, it inhibits the glycogen synthesis. It means you can see it prevents the glycogen synthesis. This is another metabolic event that is influenced by this particular hormone. Not only this, it activates enzymes for gluconeogenesis, right? Again, it is a metabolic event. It means glucagon hormones is metabolically is very important because it either activates certain type of enzymes which are essential for metabolic pathway. So here these are the enzymes which are affected by this activated form of protein kinase. Now see, if we explain in a little elaborate way about the mechanism of actions, see this is the glucagon hormone receptors, and as I've already mentioned, this is the adenyl cyclase. This adenyl cyclase helps in the formations of cyclic AMP from ATP, and this cyclic AMP is essential. As I have mentioned that there are certain types of receptors through which the hormones can act. Yes, is the receptors for glucagon hormones, and yes, in this pathway what I have seen, there is a synthesis of the cyclic AMP, and this activated form of cyclic AMP is very important for the activations of inactive pKa. Once the pK gets activated, it will alters some characteristics of the gene because it will bind in specific segments of gene that leads to the alterations of the gene expressions, which is essentials for the synthesis of a certain type of enzyme or proteins, which is metabolically important. At the same time, this glucagon hormones also acts through another pathway called through phospholipase C, and it produces the inositol triphosphates, and these inositol triphosphates along with the calcium ions promotes the expressions of these particular genes. Means what I actually want to explain here, not only the cyclic AMP PK pathway is only pathway for the actions of the glucagon hormones, it also acts through phospholipase C pathway, and in both the case it alters the expression patterns of the gene. Ultimately it produces a certain type of enzymes or proteins, and those enzymes are metabolically important for various metabolic functions of cells. Now question is, what are the important functions that is carried out by this glucagon hormone, or how this glucagon hormones and the insulin hormones helps to maintain the glucose balance. As we have already seen, the conditions that the glucagon hormones is basically secreted from specific cell types of pancreas, and this glucagon stimulates a specific metabolic events. Here it stimulates the breakdown of glycogen, and the glycogen breakdown leads to the formations of the glucose here, and ultimately leads to the changes in the blood sugar level. At the same time, and there is the high blood sugars, obviously it maintains the amount of that blood sugars from circulating blood that is maintained by insulin hormone, which is also secreted from a specific type of cells of pancreas, and you can see it stimulates the formations of glycogen. In one way it forms the glucose, in another way it forms the glycogens. At the same time, this insulin is very essential because it stimulates the glucose uptake, means the absorptions of glucose from circulating blood. In one way it is there is a rise of blood sugars, in another way it lowers the blood sugar levels. It means to maintain the homeostatic balance of glucose, obviously both the insulin hormones and glucagon hormones is equally important, right? And obviously to maintain that homeostatic balance of glucose, as both the insulin and glucagon hormones are important. Now, at the same time, this is also important. What are those factors that help to maintain the sufficient secretions of both the insulin and glucagon? See here, what is the mechanism, physiological actions of this glucagon hormone? If you see the actions, as I've already mentioned, that there is a synthesis of a number of metabolic enzymes. And those metabolic enzymes are physiologically important or metabolically important. Now we'll see how this metabolic enzymes promotes the metabolic event. See, increased blood glucose concentrations to ensure adequate energy supply during fasting or during stress conditions. It is the most important functions of the glucagon hormones. And if you see the target site of actions is the livers, and how these hormones act to the target site, like it promotes the glycogenolysis first. So here's in this particular events what we see, it stimulates the breakdown of glycogen into glucose that ultimately released into the blood. Means glucagon facilitates the formations of glucose, or it facilitates to increase the blood glucose levels. At the same time, it also equally important for gluconeogenesis. How? Because it stimulates the production of glucose from some non-carbohydrate substance, right? So what are these non-carbohydrate substances? Like lactate, glycerol and amino acids. Those are the non-carbohydrate substances from which the glucose production takes place, and this glucose synthesis or productions of glucose from non-carbohydrate substances are also influenced by the available glucagon hormone. Now, these glucagon hormones are not only promotes metabolic events, it also inhibits some metabolic events. Like there is inhibitions of glycogenesis, right? So it prevents the storage of glucose as glycogens. Generally, we know the glucagon hormone actually helps to store the glucose only, whereas insulin helps to store the glycogen. So it means there is an inhibitory role of glucagon because it inhibits the glycolysis events. It inhibits the glycogenesis events. At the same time, if you see the patterns of the glycolysis or role of glucagon hormone in glycolysis, there is an inhibitory role. It reduces glucose utilizations in the liver. That is very, very useful because it helps to preserve the glucose outputs. So not only the glucagon hormone not only involved with this type of metabolic events, at the same time, if you see the breakdown of lipid, that is also influenced by the glucagon hormones. That is basically takes place in the adipose tissue. Glucagon promotes the breakdown of triglycerides into free fatty acids as well as the glycerols. Along with this, it provides certain type of substrates. That substrates are essentials for certain type of metabolic events like gluconeogenesis. These triglycerides breakdowns ultimately produce energy. The breakdown of lipids gives the energy. And it also helps in the ketogenesis process because it promotes the formations of some ketone bodies, and ultimately this ketogenesis is very, very useful in certain conditions, mainly the fasting conditions, because in ketone bodies actually helps to provide the energy in these particular conditions. So these are some important functional aspects of the glucagon hormone. Now see what are the most important abnormalities of glucagon. Obviously, since this is the abnormalities related with the glucagon, obviously it is either due to the hypersecretions of these hormones or hyposecretions of these hormones, right? So you can see some of the terminology that is related to this glucagon abnormalities, like glucogenoma, and it is very rare conditions, and it is basically due to the abnormal mass formations of the alpha cells, right? Means the formations of tumors, and these tumors formations in these pancreatic alpha cells leads to the excessive secretions of glucagon. And there are certain symptoms that is related to this particular pathological states. These are the certain symptoms that is very much related to this particular conditions. Along with this, you can see hyperglycemia, and this hyperglycemia is very much related with the diabetes. You can see in diabetes mellitus, lack of insulin leads to unopposed glucagon secretions. Yes, what we have seen in diabetes mellitus, there is either lack of insulin or there is insulin resistance. At the same time, what we see in diabetes mellitus, along with the insufficient insulins, it promotes the glucagon secretions. Actually, there is unopposed glucagon secretions. This is another important characteristics of these pathological states. Now see, as we have already mentioned that this type of pathological states can be cured by promoting certain types of facility, like if we provide the sufficient exogenous insulin, or if we take sufficient amount of carbohydrates so that it will produce sufficient energy, glucose, and which somehow links to maintains the balance of homeostatic balance of glucose in circulatory blood. So in that aspect, we could say up to this, both the insulin hormones and glucagon hormones, in one way it is metabolically important, in another way it is pathologically is also important. Now another important type of hormones that is also secreted from a specific type of cell or pancreatic glands is called the somatostatin hormone. So the most important cell types that involves the synthesis of the somatostatin is the delta cells, right? And other than delta cells, if you see some neuroendocrine cells is also involved with the synthesis of this specific type of hormone, that is the hypothalamus, and GI tract is also involved in the secretions of this somatostatin hormone. Now, what is the most important structural features of this somatostatin hormones? Obviously, it is also made up of certain types of amino acid sequence, and you may find different segments of these somatostatin hormones. In some cases, the length will be a little bit shorter than other. You can see, in some cases, it's maximum number of amino acid sequence coordinates. In other cases, it is a little bit longer. So here, these somatostatin hormones that is secreted from the delta cells or depends on, secretions of the somatostatin hormones depends on certain factors. So what are those factors that promotes the somatostatin hormone secretions? See, these are the factors like increased blood glucose, increased amino acid and fatty acids, and increased levels of other hormones such as glucagon and insulin. These are the factors influencing somatostatin hormone. And mainly these factors influence the somatostatin hormone secretions from delta cells only. If you see the hypothalamic secretions of somatostatins, hypothalamic neurons release somatostatins into hypophyseal portal systems to inhibit the actions of certain types of hormones, like the actions of growth hormones as well as the actions of thyroid hormones, thyroid stimulating hormones. And the somatostatin hormones, which is released from GI tracts, it stimulates the presence of, it is stimulated under the presence of a certain conditions like nutrients in the GI tracts. So after this, you could say there are certain types of phase, and that phase is important for the release of these somatostatins. There are certain type of sites or locations from where somatostatins are secreted. Now, like previous ones, it is the peptide hormones. So the mode of actions is the second messenger systems, but there is a little difference from previous ones. I will explain what is the exact difference of this, the mode of actions of somatostatin hormones. I am not going into the details of this part. See here, the most important difference between the mode of actions of glucagon and somatostatin. The receptors is the same, G protein coupled receptors. and here it is known as somatostatin receptor, and it is basically distributed to certain types of organs, mainly if you see the brain tissue, you will find this type of receptors in the pancreas, pituitary, and even GI tract. And the most important things, somatostatin primarily activates the G inhibitory proteins, which is the G protein, not G stimulatory. It is the G inhibitory protein. So activations of G inhibitory protein inhibits the adrenal cycles. In previous case, it was the activations of the adrenal cycles, but here it is the inhibitions of the adrenal cycles. So inhibitions of adrenal cycles leads to the decrease in the CAMP levels. In previous case, it was the increase of CAMP level. So reduced level of CAMP reduce the activations of PK. Previous case, it was the activations of the PK, but here reduced activations of the PK, thereby inhibiting the phosphorylations as well as the downstream targets that involve the hormone actions. So in this particular case, the most important thing is the role of somatostatins in second messenger pathway is basically the inhibitory role, right? Additionally, somatostatin reduces the calcium influx. See here, up to this is okay. There is a inhibitory role of this particular hormones. Here, once the PK gets inhibited, it reduces the influx of calcium, right? It means now the amounts of available calcium is less. That inhibits the voltage-gated calcium channels, right? By inhibiting the voltage-gated calcium channels, and these inhibitions of calcium influx ultimately enhance the potassium efflux, right? And leading to hyperpolarizations. In previous case, it was the depolarizations. Means there is also changes in the membrane potentials, but here due to the efflux of potassium ion, there is a generations of hyperpolarization condition, right? And these ionic changes suppress the exocytosis of hormone-containing vesicles, and that effectively decreasing the secretions of these hormones. So what type of hormones? The secretions of growth hormone, insulin hormone, glucagon hormone. As I've already mentioned that in these slides, that somatostatins into the hypothalamic neuron release somatostatins into the hypophyseal portal systems. For what purpose? To inhibit the growth hormones, to inhibit the thyroid stimulating hormones. So now question is how they inhibit the growth hormone release, how they inhibit the thyroid stimulating hormone release. This is how they inhibit the release of both the growth hormones and thyroid stimulating hormones by suppressing the exocytosis events, right? So this is what is the general mechanism of this particular hormone actions, and at the same time you can see here's the lowering cyclic MP and inhibitions of calcium entry. Both the conditions are equally important to maintain the hyperpolarization state. And once the cell membranes gets hyperpolarized, that actually is very much essential for the regulations of this exocytosis event, right? So this is what is the general mechanism of this somatostatin hormone action. Like previous ones, the abnormalities of somatostatins also may occur under certain conditions. So these are certain conditions that may develop these abnormalities that is related to the somatostatins. So here, so up to this, what actually I have discussed from the previous class to today's lectures, I have mainly discussed three things. One is how these insulin hormones are secreted and how these insulin hormones are important, and how both the insulin and glucagon helps to maintain the homeostatic balance of glucose. And I've also discussed about what is the mode of actions of glucagon hormones as well as the mode of actions of somatostatin hormones. I've also discussed about what is the metabolic role of both the insulin glucagons as well as the role of somatostatin hormone. And at the end, I've discussed what are the different type of abnormalities that are related with these abnormal secretions of all those pancreatic hormone. So this is all about the discussions from pancreatic gland. Thank you.